Welcome to today's Explainer. We are diving really deep into the pomegranate today. And I don't just mean talking about it as a healthy snack. We're going to explore the cutting-edge biochemistry of exactly how this fruit fights cardiovascular disease, and we'll unpack the completely paradigm-shifting postbiotic known as Urolithin A.
Here is how we're breaking this down. First, we'll cover pathophysiology and clinical outcomes. Then, anatomical forms and gastrointestinal toxicity, followed by extract standardization and pharmacokinetics. After that, we'll get into the microbiome and urolithin metabolites, then enterocluster and the UCD operon, and finally, direct urolithin A supplementation.
All right, let's jump straight into Section 1, Pathophysiology and Clinical Outcomes. To really appreciate how pomegranate interventions work, we first need to understand the cellular pathogenesis of atherosclerosis, basically how arterial plaques actually form. It's a chain reaction. First, LDL cholesterol gets trapped and oxidized right inside the arterial wall. When that happens, your immune system's macrophages rush in to clean it up. But there's a huge problem. Their scavenger receptors are completely unregulated, so they just keep gorging on this oxidized LDL until they turn into these lipid-laden foam cells. Eventually, those foam cells die off, and that leads directly to plaque necrosis. Now, here's where it gets interesting. Pomegranate polyphenols actively interrupt this whole molecular cascade by directly inhibiting a macrophage enzyme called NADPH oxidase. This drastically reduces their ability to take up that dangerous oxidized LDL.
And the clinical reality of this mechanism is just striking. In the landmark Aviram cohort, patients who consumed standardized pomegranate juice daily saw an unprecedented 32% reduction in carotid intima media thickness over just 12 months. Now, keep in mind, this was a specific trial of patients with pre-existing carotid artery stenosis, but the results were absolutely remarkable. To see an actual structural regression of 32%, especially when compared to the placebo group whose arterial walls literally just continue to thicken, is a huge deal. So what is actually making this happen? Well, a major key player here is an enzyme called paraoxinase 1, or PON1. What's wild is that pomegranate polyphenols have this unique structural ability to physically bind to your HDL cholesterol particles. And when they do, they vastly stabilize and activate this highly protective PON1 enzyme. By keeping this enzyme preserved and active, the pomegranate is basically ensuring the continuous detoxification of your arterial wall, actively breaking down harmful oxidized lipids before they can cause that downward spiral of damage.
Moving on to Section 2, Anatomical Forms and Gastrointestinal Toxicity. We really need to dissect the physical fruit itself here, because consuming the wrong anatomical components can be severely toxic. The crucial point is comparing the safe, clinically validated parts, like the areoles and the juice, which contain soluble polyphenols and anthocyanins, against unrefined whole fruit powders. You see, the rind and the pith, those non-edible structures, contain massive loads of heavy, hydrolyzable tannins. When manufacturers just grind the whole fruit up into unrefined powders, the antioxidant value might look amazing on a lab sheet, but the clinical reality is that it carries a massive risk of profound gastrointestinal intolerance.
This completely explains why ingesting even a single teaspoon of raw whole fruit powder can trigger severe abdominal cramping. We call this tannin-induced mucosal precipitation. It's an aggressive mechanism. Basically, the macrotannins from the rind hit the watery environment of your gut and instantly react with the proteins in your protective gastric mucus. It literally causes a rapid chemical cross-linking. So you get this acute structural hardening that strips the protective lubrication right off your stomach lining, leaving it completely exposed and irritated.
And unfortunately, it gets worse. Compounding that stomach damage is something called visceral hypersensitivity. Humans don't actually have the enzymes to digest the high fructan loads, the FODMAPs, found in that unrefined rind. So when those hit your colon, your bacteria rapidly ferment them, producing explosive volumes of gas. If you have any kind of GI sensitivity, this sudden distension triggers an intensely painful neuroinflammatory response. This whole double whammy of mucosal precipitation and gas distension is exactly why cardiologists and gastroenterologists highly caution against raw whole fruit peel powders.
Okay, let's jump into Section 3, Extract Standardization and Pharmacokinetics.
To safely harness that polyphenolic payload without the gastric trauma, we have to look really closely at extract standardizations, because the specific chemical marker used fundamentally dictates your clinical outcome. On one hand, you have ellagic acid.
It's small, it's highly lipophilic, and it's notoriously insoluble in water. That means extremely poor human absorption. Plus, it's found in really cheap sources like oak bark, so industrial adulteration is a huge risk. But then, on the other hand, you have punicalagins, specifically punicalagins. These are massive, complex molecules entirely unique to the pomegranate, and because they are highly water-soluble, they have an amazing absorption rate of up to 95%. This is exactly why advanced punicalagin extracts represent the absolute gold standard here. Once they get inside your body, they act as a sophisticated sustained-release reservoir, continuously breaking down over many hours. Clinical trials clearly show that this targeted standardization drives profound lipid and endothelial optimization. We're talking about lowering oxidized LDL and inflammatory markers like hsCRP while physically protecting your endogenous nitric oxide to support healthy blood pressure. And best of all, it does all of this completely devoid of that nasty gastrointestinal toxicity we talked about earlier.
Which brings us to Section 4, the microbiome and urolithin metabolotypes.
Now, here is where things get genuinely fascinating. The ultimate cardiovascular efficacy you get from any pomegranate extract isn't actually determined by what you swallow. It's determined by the bacterial metropolis living in your colon. This genetic lottery definitively dictates your clinical outcome. The compounds you consume have to be transformed by your gut bacteria into powerful postbiotics called urolithins. If you belong to metatype A, your bacteria produce urolithin A, which is highly cardioprotective. But if you're metatype B, your gut produces urolithin B, which is actually a biomarker for advanced metabolic dysfunction and statin resistance. And if you are metatype 0, well, you're a non-producer. You completely lack the necessary bacteria. For those folks, generic pomegranate interventions will just fail entirely. So naturally, this begs a massive question. What exactly is the precise bacterial mechanism that takes these inert parent compounds and transforms them into highly potent, cardioprotective postbiotics?
Let's uncover that in Section 5, Enterocluster and the UCD operon.
This was a profound biological mystery, and it was literally just solved by cutting-edge transcriptomics in 2024 and 2025. Researchers discovered that the final transformation into cardioprotective urolithin A is executed exclusively by a very specific subset of Enterocluster species in the human gut. But here's the catch. Merely harboring these bacteria isn't enough. They must actively transcribe a precise, highly specialized genetic sequence known as the UCD operon. Only then can they build the immensely powerful enzyme complex required to get the job done.
This sequence of events is just an extraordinary tale of evolutionary microbial warfare. Urolithin A is absolutely not some passive byproduct of human digestion. When an intermediate compound called urolithin C arrives in the gut, it violently binds up all the free elemental iron. Suddenly, these Enterocluster species are facing severe, life-threatening iron starvation. In a desperate move to survive, the bacteria massively upregulate transcription of that LUCD operon we just mentioned. The resulting enzyme cleaves a specific hydroxyl group off the urolithin C, converting it into urolithin A. And because urolithin A completely lacks the ability to bind iron, the bacteria save themselves from starvation. As a byproduct of their survival, they release this incredible cardioprotective urolithin A right into your bloodstream.
All right, let's wrap this up with Section 6, direct urolithin A supplementation. Because such a massive portion of the population lacks that active operon, modern clinical pharmacology has pivoted to a fundamental paradigm shift. By bypassing the microbiome bottleneck entirely, direct urolithin A supplementation acts as a highly penetrating intracellular signaling molecule. Its primary, incredibly powerful mechanism is mitophagy. This is the forceful cellular recycling and clearance of aged, dysfunctional, and toxic mitochondria from deep within your cells. And this is absolutely crucial, because severe mitochondrial dysfunction is a foundational hallmark of cardiac decline.
When you literally force the myocardium, your heart muscle, to clear out and recycle these defective mitochondria, it leads to profound mechanical recoveries. It actively reverses cardiac aging. We are talking about vastly enhancing systolic contraction strength, drastically improving how the heart relaxes during diastole, actively preventing pathological enlargement of the heart chambers, and successfully preserving ejection fraction. I can't stress this enough. Finding a single intervention that improves both systolic and diastolic functions simultaneously is incredibly rare. And this culminated in a pivotal 2025 clinical breakthrough.
Human patients taking 1,000 milligrams of direct oral urolithin A daily for 4 months saw a massive reduction in circulating plasma ceramides. Ceramides are these highly atherogenic lipids that strongly drive systemic insulin resistance and coronary artery disease. By lowering them, urolithin A profoundly lowered the CERT score. This is a clinically validated biomarker that accurately predicts future cardiovascular events and overall mortality. It proves we can now deeply alter predictive cardiovascular risk without relying on the unreliability of human gut bacteria.
So, to really cement all of this, let's compare these interventions to see the exact bottlenecks. Pomegranate Juice has incredible data, like that 32% regression, but it relies heavily on your microbiome. Whole peel powders trigger such severe GI toxicity that it completely negates any systemic benefits. Punic extracts heavily and safely optimize lipids, but they still demand that your bacteria have that specific UCD operon. Direct oral urolithin A, however, emerges as a massively reliable, scientifically rigorous therapy, simply because it removes the biological bottleneck entirely.
Which leaves us with a pretty major question to consider as you map out your own evidence-based cardiovascular interventions. In the clinical pursuit of extreme longevity and cardiovascular resilience, can we afford to gamble on the genetic lottery of our gut bacteria? Or is it time to bypass the microbiome entirely? Thank you so much for joining me on today's Explainer. Keep learning and keep looking at the science.